I spoke about molecular drivers and therapeutic targets of lung cancer transformation. So to speak, I spoke exclusively about EGFR-mutant lung adenocarcinoma that transformed to small cell lung cancer as a mechanism of resistance to tyrosine kinase inhibitors. This phenomena has been noted for almost a decade ago. This was first reported in 2011, and since then it had been reported by various groups. In our study, we, for the first time, did a multiomic analysis on what we called a combined histology tumors, that we had 11 cases where the lung adenocarcinoma component and the small cell component were in the same spatial region. We performed microdissection of these samples, and we analyzed them with various platforms like whole exome sequencing, bulk RNA sequencing, methylation analysis, and immunohistochemistry. And the results really gave us a lot of insight into these very understudied tumors. We have known about this phenomenon, but it hasn't been studied so far. So what our genomic analysis, or the analysis with whole exome sequencing, told us is that p53 and RB1 loss was one of the most characteristic feature of these tumors. So almost all of them had a loss of function mutation in p53 and RB1, or RB1 was functionally lost in these tumors. And so that seems to be one of the main genomic characteristics. And what we learned from RNA-Seq analysis is that several pathways gets either turned on or turned off during this process of transformation. And some of the important targets that I would like to point out is that we observed Notch inhibition, and we also saw upregulation of several PRC2 complex genes. We saw upregulation of DNA damage response genes as a sort of a predictive pathways that could predict these tumors that are more poised to transform. And we also saw a suppression in the anti-tumor genes. So we found several immune pathways to be suppressed in these tumors that were transforming to small cell lung cancer. So I think in terms of the pathways that we find the most interesting and that we are going to follow up in future studies would be the AKT signaling pathway, the Wnt signaling pathway, and several epigenetic modulators. And I think one of the more characteristic features is that the suppression of the anti-tumor immune response, which we are definitely going to follow up in further studies. In terms of therapeutic targets, we found AKT inhibition to be effective in an EGFR-mutant mouse model, where we found that when you combine AKT inhibition with Osimertinib, which is a standard therapy for these EGFR-mutant lung adenocarcinoma, the combination actually performed better in our in vivo studies or in our mouse models than either of the single agents alone. So that is one of the therapeutic targets that we have identified. I'm sure there are many. It's an active area of investigation, and I would be very interested to see what comes up in future studies. But I think this study is important because this not only gives us an insight into the biology of these tumors, but it also points out towards several pathways and targets that could be followed on in future studies, and also additional targets that I think needs investigation further.